This is my lab in University of Tehran, and most of PhD students and Master of student come from different subjects, not chemistry, biology, and engineering, because my subject is the nanobiotechnology. But before that, I talk about this, my topic title and research. My research topic is cancer biomarker detection, pathogenic bacteria detection, and virus detection, and wearable biosensor. Before that, Professor Hassan talk about this basically about the biosensor and sensor, and sometimes about this is which have a signal and other ways. First time, let an overview of cancer start is the worldwide. Cancer continue to be a major global of concern worldwide. As you can see, about 10 million deaths in the 2020 in worldwide. And it is important, this 70% mortality rate occur in the world, low and middle income. And other things is each year about approximately 400,000 children recognized as new cancer for just children in worldwide. And other things is the cancer is a second cause of mortality. Is after that the heart disease. It is important and it is interesting, risk factor and mortality rate is different between male and female. In the fatality in male and female, it's 30% and 90% respectively. It is important for us, how can reduce this mortality rate? Cancer can be, it is possible we try to the mortality rate, yeah. As you know, we have a yearly detection cancer is very important. Yearly detection cancer have a two key component, yearly diagnosis and screening. As you know, this yearly diagnostic consists of the first time recognize a symptom. After that, we need to the treatment. After that, we need to the timely treatment. Timely treatment after that is yearly diagnostic is very important. On the other hand, screening, as you know, is screening. We use a simple test. For just is the population healthy before a symptom is appear. This is the same as you know, some is for example mammography, for example some other tests, as you know, for screening, because before a symptom appear, you can recognize the symptom. We try to together is the yearly diagnostic and screening help us to reduce a mortality rate. We need this instrument to recognize an accurate medical device will help us to precise diagnosis and treatment rapidly. This instrument is very important for us. This is the same, basically analytical chemistry, fluorescence immunoassay, chemiluminescence immunoassay, and ECL, electrochemiluminescence. The most of them, this company use these techniques, and most of them, the teaching is a student for this approach for detection target. This is as you can see, is the roadmap. Just one instrument. This is the subway map, but it's just a different antibody to detection of not this cancer, heart attack, thyroid, and other biomarker, bone marker. Just one instrument recognize all of them symptoms, as you can see. This instrument very good and high repeatability and reproducibility and accuracy is very high. But unfortunately, this instrument very expensive and need to the expert operator. We cannot access, for example, low and middle income access this instrument. Very, very expensive for us. We need to try to the alternative option for this. As you can see, is better for us is the biosensor help us to find if this is something. Is the better. Is the, as you know, is the biosensor the analytical device combined in the biological components such as enzymatic, aptamer, DNA, and other biological components. Biosensor is the incorporated with nanomaterial, help us to enhance the sensitivity. The new generation biosensor is the nano-biosensor because we use nanomaterial. Nanomaterial help us to the high sensitivity, because the better for us, the immobilization is better, is easy, and the transduction is better and very, very the fast electron transfer, and help us to enhance in the sensitivity. So we need to choose the alternative option because the instrument is very expensive. The biosensor as the alternative option. This, if we try to my lab, try to produce and develop a new biosensor based on the nanomaterial. As you can see, this is the different nanostructure synthesized by lab. This is the recently. As you can see, is the different sheet, two-dimensional sheet. Is the graphene and the carbon nitride and the bottom is the Maxine nanosheet. This is the nanoparticle, different nanoparticle has synthesized my lab. Gold nanoparticle, gold nanorod, IORG or IOflower, nanofiber, MOF or metallic organic frame, nanocluster and triangle or premise is the triangle is the for base is the nano-premise is the gold triangle, silver triangle is synthesized in my student. The first, what is the cancer biomarker? As you can see, this is the DNA, RNA, and cancer cells. The cult is the cancer biomarker. When help us to find catching early cancer. This is cancer is the like a small hint to us. What happened? This because useful information. The cancer, how is growing, how is recurrence, and maybe it is useful is what happened, the cause to cancer. This is the cancer biomarker is different category: RNA, DNA, cancer cell, enzyme, and protein, and some stem cell, and other biomarkers is different. The first step in my PhD student focus in the cancer biomarker name is the epigenetic biomarkers. Epigenetic refers to change to the gene expression. Gene expression change. The epigenetic refer to this biomarker is the main category is the cancer biomarker. What happened? This genes named as tumor suppressor gene. When gene is in activation, the methylation, the CPGI land can occur. When CPGI land is a hypermethylation,